Welcome back to the deep dive. Today we're jumping into a stack of recent reports you've shared, focusing on some really interesting developments in cancer research and potential new treatments.
That's right. We got four sources here covering different angles, new drug candidates, radiopharmaceuticals, and even some basic science on how cancer cells resist therapy.
Right. So our mission today is to unpack these together, pull out the key information, and sort of understand what it all might mean.
Absolutely. We'll start with the agents that are further along, the ones in clinical trials, and then we'll look at a fascinating preclinical finding.
Okay, sounds good. Let's start with those clinical trial updates. First source, ATRN-119. It's called an ATR inhibitor. What does that mean exactly?
Okay, so ATR is a protein, and it's a really critical part of how cells repair damaged DNA. Cancer cells, because they grow so fast and have lots of mutations, often have other repair pathways broken.
Yeah.
So they become really, really dependent on ATR to survive.
Ah, okay. So inhibiting ATR, like with ATRN-119, is hitting them where they're vulnerable.
Exactly. You're taking out a key survival mechanism. And this specific drug, ATRN-119, the source mentioned its unique structure, helps it be very selective just for ATR, which is good for, minimizing side effects.
And it seems particularly relevant for tumors with certain mutations already, specifically ATM loss of function.
Yes, that's the synthetic lethality idea. ATM is another DNA repair protein. If a cancer cell already lacks working ATM, its main repair route is faulty.
Right.
So then you block the backup ATR with this drug. The cell just gets overwhelmed with DNA damage and dies. Healthy cells, though, they still have working ATM, so they can usually cope.
That makes sense, finding that specific weakness. So what did the early trials show?
Well, the source highlighted data from 3 patients on a specific dose, 550 milligrams twice daily. They saw tumor shrinkage of 7%, 14%, and 21%.
Okay, maybe not huge numbers on paper, but why is that considered notable?
Well, a couple of reasons. First, this happened before they even reached the dose they planned to use in the bigger phase 2 study.
I see.
And second, the drug was used all by itself monotherapy. Seeing that kind of activity alone relatively early on is definitely encouraging.
And what about safety? That's always crucial.
Big point here. The source emphasized minimal hematological toxicity that's damage to blood cells and bone marrow. This is often a problem with other drugs in this class. If ATRN-119 really is easier on the bone marrow, that's potentially a huge advantage. It might allow for continuous dosing and, critically, combining it with other drugs like PRP inhibitors, which can also affect bone marrow.
Right, that opens up combination strategies. And how common are these ATM mutations it targets?
The source mentioned they're found in up to 24% of solid tumors. So if this pans out, it could be relevant for a pretty significant group of patients.
Okay, definitely one to watch. Let's move to the third source. This one introduces SN1117-beta DTPA. Completely different approach, it seems, radiopharmaceuticals.
It is. And it's what we call a theranostic.
Theranostic. Therapy and diagnostic rolled into one.
Exactly. One agent does both jobs. It helps doctors see where the cancer is, and then it delivers targeted radiation right to that spot. SN117-beta DTPA is being looked at for painful bone metastases, especially in metastatic castration-resistant prostate cancer, MCRPC.
So it finds the bone spots and treats them. How does it work?
Okay, so the SN117-meter part is the radioisotope radioactive tin. The DTPA is like a delivery truck, a chelating agent that carries the tin and is attracted to areas of high bone activity, like metastases.
Right.
For imaging, the SN117 meter gives off the gamma photons, which standard hospital cameras can detect, so doctors can see, yes, it went to the bone lesions.
That's the diagnostic part.
Yeah.
And the therapy.
Once it's sitting there in the bone metastasis, the SN117 meter decays and releases these low-energy electrons, conversion electrons, and auger electrons. The key thing is they have a very, very short range in tissue, like 0.2 to 0.3 millimeters.
Tiny range. Why is that good?
Because it means the radiation dose is super concentrated right there, hitting the cancer cells. But it doesn't travel far enough to significantly damage surrounding healthy tissue, especially the bone marrow. That's often the limiting factor with other radiotherapies.
Less collateral damage, essentially. What's the clinical trial status?
There's an active phase 2 trial looking specifically at pain relief in MCRPC patients, and a phase 1 trial looking at safety, dosing, and maybe even some anti-tumor effect in bone mets from prostate, breast, and lung cancer.
And past data.
Yeah, earlier phase 1-2 trials with over 120 patients showed pain relief for most, and about 30% had complete pain resolution. Relief started fast, within days, and could last over 3 months. And again, minimal bone marrow issues reported.
That sounds really valuable for quality of life. Is it just about pain, though?
Well, initially, yes, mostly palliative, but the source notes the research is shifting. They're now exploring if it can actually fight the metastases themselves, not just ease the pain. That's a big step.
Moving from symptom control to potentially disease control.
Got it. Okay, fourth source, also prostate cancer related, OncoACP3.
Yes, another targeted approach. This one's a small molecule designed to stick to something called acid phosphatase 3, or ACP3. It's an enzyme that's found in much higher amounts on prostate cancer cells compared to normal cells.
So another way to target the cancer specifically.
Exactly. And like the last one, it's also being developed as a potential theranostic for imaging first, then therapy.
What did the early imaging studies show?
Preliminary phase 1 data using it for PET imaging looked good. It showed the molecule was taken up selectively by the prostate tumors, it stayed there over time, and importantly, not much accumulated in healthy tissues. That's what you want to see.
Good targeting. So the next step is therapy.
Right. Based on that imaging data, confirming it goes to the right place, they're planning a trial using the same molecule but attaching a therapeutic radioisotope to it.
And what kind of radioisotope does it matter? It does. The source highlights this is compatible with alpha emitters, like actinium-225. Alpha particles are different. They travel an even shorter distance than those electrons we just talked about, like micrometers, the width of a few cells.
Wow, even more localized.
But they pack a much bigger punch. They deliver a lot more energy in that tiny space. So the idea is a very potent, localized kill of cancer cells right next to the molecule, with potentially even less damage to nearby healthy cells.
High energy, short leash. The source mentioned a licensing deal, too.
Yes, it did. And that kind of business activity reflects the growing excitement and investment in this whole radiopharmaceutical area. There's a clear need for better diagnostics and therapies that are more effective, but also less toxic,
Yeah.
especially for common cancers like prostate.
Okay, so two promising radiopharmaceuticals alongside the targeted oral drug. Now let's completely switch gears to that preclinical discovery in the second source. Cholesterol and heat resistance. That sounds pretty different.
It really is, and quite fascinating. This study out of Japan found that cholesterol, stuff in cell membranes, helps some cancer cells survive high temperatures. This might explain why hyperthermia heat therapy doesn't work on all tumors.
Hyperthermia. That's heating the tumor up to kill cells, right?
Exactly, typically around 50 degrees Celsius. The heat damages cancer cells, often more than healthy cells. It can also potentially help the immune system recognize the cancer better, so it's interesting alongside immunotherapy. But yeah, some tumors just shrug off the heat.
And cholesterol is the key to that resistance.
Seems to be a major factor, yes. The researchers found these heat-resistant cancer cells had way more cholesterol packed into their membranes compared to the heat-sensitive ones.
How does extra cholesterol protect them from heat?
Well, heat tends to make cell membranes more fluid, sort of looser. If they get too fluid, they can break down, and the cell dies. The extra cholesterol seems to act like a stiffening agent, almost like a shield. It keeps the membrane stable even when it gets hot, preventing that heat damage.
Huh. Like reinforcing the walls. Did they try removing the cholesterol?
They did. They used cholesterol-depleting drugs. Think drugs similar to statins people take, both on cells in the lab and in mice with tumors.
And the results?
Pretty dramatic. Lowering the cholesterol made the cancer cells much more sensitive to heat. In the mice, combining cholesterol depletion with heat therapy caused significant tumor shrinkage, sometimes even complete disappearance. Much better than heat alone.
Wow. Using a common drug type to boost a physical therapy, what could this mean for patients?
Several things. First, maybe measuring tumor cholesterol could predict who will respond well to hyperthermia. Second, maybe you could actually treat patients with cholesterol-lowering drugs alongside hyperthermia to make it work better, even against resistant tumors.
And there was an immunotherapy action mentioned.
Right. The way cells seem to die from this combo therapy, cholesterol depletion plus heat, is often necrosis. Necrotic cell death tends to be good at alerting the immune system. So this whole approach might actually help make immunotherapies more effective too.
So understanding something basic like membrane cholesterol could potentially improve both heat therapy and immunotherapy. Amazing.
It really shows how fundamental research can open up completely new treatment avenues.
So looking across all these, the targeted drug, the theranostics, this cholesterol, finding what's the big picture here? What connects them?
Well, they all point towards increasingly precise and tailored ways to fight cancer, whether it's exploiting a specific gene mutation like with ATRN119, using molecules like SN117-DTPA or OncoACP3 to guide radiation directly to the tumor,
Uh-huh.
or even understanding how to manipulate basic cell components like cholesterol to make existing therapies like hyperthermia work better. The common thread is hitting the cancer hard while sparing the healthy tissue as much as possible.
More precision, less toxicity. That seems to be the driving force.
Absolutely. It's about understanding what makes cancer cells different, their genetics, their proteins, even their membranes, and using that knowledge to design smarter therapies, making treatments more effective, better tolerated for more people.
It's definitely clear there's a lot of innovation happening, pushing for better ways to tackle this disease.
No question. Each of these studies, clinical or preclinical, adds a piece to a very complex puzzle.
So as you think about these different strategies, targeted drugs, guided radiation, overcoming resistance mechanisms like heat resistance, it's interesting to consider how they might eventually work together, perhaps in combination, to treat cancer more effectively in the future.
That potential for synergy, combining different approaches, is definitely where a lot of future research will likely focus.
Stay strong and fight on.